Perfect. Thank you, Dan. So as you said, we'll now be moving on to the Q&A portion of today's webinar. Many of you have already entered your questions, but if anyone else would like to ask Dan a question, please go ahead and enter it into the Q&A box now. Dan, the first question is, have you noticed a significant effect caused by the latency of the micro LED ramp time?
So that question, that's a really good question. So this, I assume that means from the time, let's say your signal generator outputs the voltage, to how long does it take to the micro LEDs to light? And that is essentially, it's so short you can dismiss that delay. So the LEDs light almost, in a time scale that's much faster than the physiological time scale. So it's on the, I haven't been able to measure it, but it's on the microsecond, hundreds of microseconds at most, I would guess. That would be a better question for USIC, but it doesn't come into play in terms of how fast you can stimulate.
Thank you. Next question. The amount of power measured by the micro LEDs is small compared to optic fibers, e.g. Microwatts from the LED compared to milliwatts from the fiber. Does this affect the behavioral response?
So that's a really interesting point, right? So optogenetics is used for many things at very different scales. And so if you're looking to control the behavior of an animal, I mean, in class, some of the early classic experiments from people like Carl Diceroth, they stimulated dopamine neurons and with optogenetics and showed that you could get place preference, stuff like that. That's not what the micro LEDs are designed for, and that's not their forte. So obviously, if you want to turn on all dopamine neurons in the brain, or light up the whole hippocampus and turn the whole thing on or off, you would need an optic fiber to do that. But in reality, if you have a light source such as the micro LED close to a neuron, you're probably 100 microns or less from the cells you're recording, you actually need, you don't even need that, you need very, very small amount. Found Wu and around Stark, they actually quantified the minimum amount, but in our hands, it's definitely 100 microwatts. It depends. So this actually depends on the cell type, but it definitely is always sufficient to drive the neurons that you're recording. But if you're looking to affect a large number of neurons to control behavior, this is not the right. In my opinion, this is not the right tool. So in fact, there isn't necessarily an overt behavioral response to stimulating this few neurons.
Okay. Another attendee states, I've seen in some papers a radiant flux measurement included. Is this measuring concerned with light distribution within tissue? And if yes, how?
So I'm not exactly sure of the metric or what kind of figure they're imagining, but the tissue does disperse light. Obviously, different wavelengths are different. It's not, the brain isn't made of water, right? There's proteins and everything that are affecting the flow, the movement of light. The distribution of the light in space and in an agar, which is more similar to the brain than air, is quantified in in terms of just light itself spreading in the brain. There's a 2012 paper from Aran Stark and Yuri Bushaki in Journal of Neurophysiology that really nicely looks at the spread from small etched fibers. And I think in the paper from Fan Wu and Aran Stark, Yusik, and Yuri with the micro LED probes. I don't know off the top of my head. And is the measurement attributed to the specifics of the LED? The LED driver controller doesn't have anything to do with it. It would be the color of the LED and the shape of the emitter, I guess, exactly. So in general, the blue light is going to not travel as far as, let's say, red light.
Okay. Next question. What is the smallest number of neurons you can stimulate?
So that's why I included that plot. So in CA1 in the hippocampus, if all the pyramidal cells are expressing channelrhodopsin, it's hard to reliably stimulate a single cell. So it's it's like a handful of cells is really the smallest thing, and the biggest thing that we need to bring that to a better number, to 1 obviously would be the best number, is the fact that the opsin, the biggest problem is it's not with the micro LEDs, it's with the opsins themselves. So the micro LEDs are great and are perfect in terms of the fact that they can light up a tiny amount of tissue. But the problem is within that volume of tissue is going to be not just one cell and not just the soma of one cell or the soma of two cells, but it's also going to be the dendrites and the axons, all of which express channel rhodopsin along the surface. And so a really big deal is and something that we're waiting for, and I know lots of people are working on, is generating opsins that are expressed exclusively in subcompartments of the cell, such as the soma, which would enable you to lower the number of the smallest number of neurons.
Okay, next question. Do all neurons have the same threshold for light stimulation?
So the answer to that is no, even within the hippocampus. And so interestingly, the pyramidal cells, so if you express channel rhodopsin in that it's an AI32 mouse, if you cross that with a Cree mouse that drives the expression in interneurons or pyramidal cells, they have very different thresholds for light. So it turns out actually the interneurons have a higher threshold. They actually need more light, which may sound paradoxical because they have higher firing rates, a different input resistance, et cetera. But that's a repeatable effect, and it probably has to do with the expression of the opsin.
Great. One attendee notes, thanks for the great talk. Can you elaborate on the method of computing the transition probability, specifically definitions of the P causal, P fast, and lambda slow? What is the ground truth?
That's a question that I would be easier to, or more effectively answer in a offline or one-to-one session. So they can feel free to email me.
Okay, well then that is all of our questions that we have. So thank you. That concludes our webinar today. Don't forget to register for next week's webinar in our series, Pre-existing Hippocampal Network Dynamics, Constrained Optogenetically Induced Place Fields. You can find registration links at plexon.com and can contact Plexon at info@plexon.com if you have any questions about purchasing the NeuroLight optoelectrodes. Thank you.